My name is Katie and this is my dog Caysie. Today, I wanted to describe a novel bioinformatics system that I created which can aid researchers in developing new medicines to treat any disease pathogen. A few definitions first. Bioinformatics is the use of computers, software, and mathematical models to process and integrate biological information from large data sets. Computational biology is the process of analyzing and interpreting biological data. These are two new fields that grew out of the Human Genome Project. Why are proteins important? Well, as explained in my previous video on bioinformatics and computational biology, proteins are the main agents of biological function. Therefore, it follows that proteins should be a key interest in molecular-based studies of biological disease states. What are protein-to-protein interaction or PPI maps? Well, a PPI map is simply a network map showing all known interactions between proteins. This is an example of what one may look like. Why are PPIs so important? Well, determining the molecular mechanisms that lead to disease states can be an even bigger challenge than deciphering the genetic basis of the disease itself. For example, in the case of Huntington's disease, the culprit gene and protein was identified in 1993 as HTT. But it wasn't until 2004 that Goler et al. Mapped all the PPIs in Huntington's disease and discovered that the interaction between HTT and GIT1, a GTPase-activating protein, mediates HTT aggregation. Once this knowledge was obtained, better treatments could be formulated. Through the years, information from protein interaction experiments was put into publicly available databases containing biological information. These are some of the databases I used in formulating my overall process. Consult genome databases for proteins from species of interest, consult interaction databases, and create a protein-to-protein interaction map, or PPI, of these species, perform network analysis on the PPI map to find key nodes or proteins in the network, and consult drug and chemical databases to find candidate compounds for pharmacological research that affect those proteins. Next, to automate my overall process, I created a new bioinformatics system which can be run on a laptop or desktop computer. I call it the BioAssistant. The BioAssistant automates my overall process, shown earlier, integrates other bioinformatics systems from around the world via the World Wide Web, searches for protein-to-protein interactions for bioinformatics databases on the internet, creates a PPI map and aids in network analysis, and suggests drugs and chemical compounds that target a given protein. It also provides a fun, flexible, and interesting interface that assists researchers questing for chemical compounds targeting a given protein, and thus the pathogen. The next slide contains a block diagram of the BioAssistant software. It is written in Java. The pink blocks are my original code. It is able to access any web service which supports the PSIC QUIC interface, including BioGrid, Bind, and others. Let's take a look. This is an overall block diagram of my BioAssistant software. It interacts with the user who can use natural language. This component is called the BioAssistant controller. Its responsibility is to control and interact with the other components in my system. This component is called the user command decider. Its responsibility is to take input from the user and determine which command is requested. In order to do this, it makes use of other natural language processing software that I wrote. This component is called the interact getter and writer. Its responsibility is to interface with other bioinformatics systems on the World Wide Web, collect protein-to-protein interaction data based on the user's command, and save it for future analysis. This component is called Analyze Interactions. Its responsibility is to help the user perform network analysis on the protein-to-protein interaction data obtained earlier. In order to do this, it creates a PPI map and aids in network analysis techniques by making use of an open source software called Cytoscape. This component is called the Drug Bank Searcher. Its responsibility is to search a drug and chemical database for candidate chemical compounds for pharmacological research that affect a given protein. Let's take a step back. For testing and demonstrating my BioAssistant, I use the HIV-1 virus. This is the virus which causes AIDS. In general, a virus is comprised of DNA or RNA surrounded by a protein coat. In the case of HIV, we have a retrovirus. Shown here are the surface glycoproteins. These bind to the host cell. This surface glycoprotein is known as EMV or GP160. It cleaves into two groups, GP120, shown here, and GP41, shown here. This is a human HIV-1 PPI network that I created to perform network analysis on. It serves as an example as to how a researcher may perform network analysis on a PPI. The HIV-1 proteins are shown in yellow, and the human proteins are shown in green. Given the topology of this human HIV-1 PPI network, the network analysis characteristic of most importance is the degree centrality, which is the number of connections a node or protein has to other nodes. As can be seen by this diagram, ENV or GP160 has the highest degree centrality and connects to many human proteins, and therefore is most important to viral function. So large hub proteins may be interesting targets to obstruct viral function. Now for a live demo of my BioAssistant in action. Here is the live demo of my BioAssistant software. Here I am running BioAssistant. And here, BioAssistant is printing its greeting and telling you what it is capable of. It can use computational biology to help you find new chemical compounds or drugs to treat disease. So, it is asking me what I want to do. I will reply with a natural language sentence. Please investigate interactions between human and HIV-1. Now, the user command decider component of my code is parsing my sentence and determining what my command is. Now, the interact getter and writer component of my code would search other bioinformatics databases on the World Wide Web for protein interaction data. In this case, I've configured it to use Intact, a database which happens to be in Europe. Now, as you can see here, the analyze interactions component of my code is aiding in network analysis by constructing a PPI map of the data obtained earlier. It does this by making use of an open source software called Cytoscape. Now here I am changing it to bio layout. This makes it easier for network analysis, and this network may look familiar to you because it is similar to the protein-to-protein interaction network that I showed in a previous slide. And I used that network as an example of how researchers may use network analysis techniques on such a network. As I explained in that slide, large hub proteins may be interesting targets to obstruct viral function. And I see here a large hub protein. What is this protein in the middle? This protein is ENV. So let's use BioAssistant to search for chemical compounds that target ENV. Search for chemical compounds that target ENV, if you please. And you will notice that the drug bank searcher component of my code has searched data from a drug and chemical database supported by the University of Alberta in Canada, known as Drug Bank, and it has found candidate chemical compounds for pharmacological research that target ENV. I would like to point out that BioAssistant can be used for any given pathogen. So, let's consider a different pathogen, say H1N1 or the swine flu virus. Please investigate interactions between human and H1N1. Now BioAssistant is going out on the web.
Now I am changing it to bio layout. And you can see that this PPI network is for human and H1N1. Here, researchers could perform network analysis on this PPI, select one or more proteins of interest, and use BioAssistant to search for chemical compounds that target these proteins.
In summary, I created the BioAssistant, a novel bioinformatics system that automates my overall process, providing assistance to researchers in finding new medicines to treat any disease pathogen. In the future, I plan to make the BioAssistant code publicly available. The BioAssistant performs the following. It utilizes data from gene-to-protein databases, constructs a protein-to-protein interaction map, or PPI, and aids in network analysis, and it queries chemical and drug databases to find candidate chemical compounds which target specific proteins. I hope you found this talk to be of interest. Bye!